Hello. Slide is visible to you, all of you?
Yes, sir.
Slide is
Yes,
visible or
sir.
not?
Yes, sir.
Okay. So first I will start with the carbohydrate digestion, absorption, and metabolism. You have studied in Unit 1, I think almost 1 year before. So just to recapitulate that things, I will be going through it very hurriedly. You just try to remember the things. Okay. So first thing is the classification of the carbohydrate. Classification of carbohydrate, you have to remember that mostly that classification of carbohydrate based on that unit, how many units of glucose are there, monosaccharides, oligosaccharides, and the polysaccharides. People generally ask the questions from the oligosaccharides and polysaccharides. Particular, suppose if you are asked that raffinose is a monosaccharide or oligosaccharides or polysaccharides, so you have to tell that it's a raffinose means it's a 3 unit of sugars, glucose, fructose, and galactose. So definitely it will be coming under the oligosaccharides category, because if any particular carbohydrate contains 2 to 10 units of glucose, so they are considered as oligosaccharides. So trisaccharides examples are raffinose, kestose, isokestose. So because they contain 3 glucose units. Disaccharides means definitely there will be 2 glucose units. Sucrose, that's the table sugar, so it does contain glucose and fructose. Lactose, it's milk sugar, glucose and galactose. Maltose, again it is consist of glucose and glucose. Like that way you have to remember the different carbohydrates. Tetrasaccharides and pentasaccharides are very unique. You have to remember the example of tetrasaccharide and pentasaccharides. So example of tetrasaccharide is tachyose, and example of pentasaccharide is verbascose. So verbascose and tachyose, what does it contain? You see, raffinose contains glucose, fructose, and galactose. So along with that glucose, fructose, and galactose, one extra galactose unit will be there for the stachyose. Someone wants to join? So that is the stachyose, and pentasaccharides, that there are 5 sugar units, that's the verbascose. So along with that stachyose, if one more unit of galactose is attached there, that will be the verbascose. So another one is the polysaccharides. You know these things, homopolysaccharides and heteropolysaccharides. You have to remember cellulose is a homopolysaccharides because it's the single glucose unit. Starch, dextrin, glycogen, all are coming under the homopolysaccharides category because there is only single glucose units, that is the glucose only is there. Other than that, there are no other monosaccharides. So similar type of monosaccharides is attached there. That's why it is the homopolysaccharides. So you have to remember cellulose, starch, dextrin, all are coming under the homopolysaccharides category. Whereas the hemicellulose, which is also a structural polysaccharides, it is not a homopolysaccharides. In the hemicellulose, there are glucose, arabinose, xylose. There are so many glucose attached in that particular hemicellulose molecules. So that's why it's a heteropolysaccharides. One important thing is that you have to remember, very frequently asked question is that starch and glycogen specifically. Starch is the reserve carbohydrate for plant, whereas for animal it's the glycogen. So starch, again there are 2 types or 2 different form. One is the amylose and another one is the amylopectin. So content of amylopectin is more, almost you can say 60 to 70%, whereas the amylose is hardly 30 to 35%. Some students are still joining. Let me allow them to get in. Okay. So here you know. So this is the classification of the carbohydrate. Starch, as I have told, the difference between amylose and amylopectin. What is that one? Amylose, there is glycosidic linkage where it is alpha 1,4 glycosidic linkage. But for amylopectin, what is the difference? Amylose and amylopectin, in the amylopectin, there is a branch chain of alpha 1,6 linkage. Now, here you have to remember, if the glucose molecules are attached by alpha 1,4 glycosidic linkages, mammalian enzymes can hydrolyze that particular carbohydrate, or that can break that alpha 1 4 linkage. That's why all of us can digest this starch, which is mostly present in the cereals, what we are taking daily. So we can digest this because of this glycosidic linkage, which is of alpha 1 4 type. But if it is beta 1 4 type, which is there in case of cellulose. So if it is beta 1 4 type, in that case mammalian enzymes cannot digest that things because they cannot break that beta 1 4. So cellulose cannot be digested by mammalian enzymes. It is done by the enzymes synthesized or secreted by the microorganisms. Is it clear? Are you getting me? Okay. So this is the difference between starch, that is amylose and amylopectin. And glycogen, as I have told, this is the stored carbohydrate in animals. And this glycogen, you should remember, glycogen is similar to that of amylopectin. What is there in the amylopectin? There are alpha 1 4 straight chain, and there is a branch chain of alpha 1 6. The glycogen is similar to that of amylopectin. You have to remember that thing. Then coming to the overview of digestion in case of monogastrics of animals, how the carbohydrate get digested in monogastric species, including the human being. In mouth, there is salivary amylase, which is responsible for digestion of the starch. In stomach, there is no enzymes responsible for carbohydrate digestion. In small intestine, again, it is the pancreatic amylase. Then other than that, there are several brass border enzymes which is responsible for the digestion of disaccharides. Suppose if I am talking about the sucrase, it can digest sucrose. If I am talking about the galactase, it can digest the galactose. If I am talking about maltase, it can digest the maltose. So all these sucrase, maltose, lactase, all these things are there in the small intestine in the brass border. In the large intestine, as such, there is no enzyme secreted by the mammals, but there are some bacterial populations, that means microbial populations are there, and they can digest that particular carbohydrate, basically, which is structural in nature, structural polysaccharides, by the microbial populations. So even whenever we are also taking some leafy vegetables, if you see the logic that leafy vegetables means definitely there will be some structural polysaccharides, there will be more crude fiber, whether we can digest it or not. Yes, we can digest it because there are some microbial populations in our large intestines which is responsible for the digestion of these structural polysaccharides. But mostly it is the mouth where there is salivary amylase, then in small intestine, pancreatic amylase, and other disaccharides are there. So here, what will be the end product of carbohydrate digestion in case of non-ruminants? It is the sugar unit, that means glucose, fructose, galactose. And if we compare it with ruminant species, what is the difference between these two species? I mean, non-ruminant and non-ruminant species. Here, in case of ruminant species, you won't find glucose as an end product of carbohydrate digestions. What will be there? There you will be finding volatile fatty acids and gaseous products. So questions you may get that what will be the end product of carbohydrate digestion in ruminant species, whether it is glucose or something else, that is volatile fatty acids and the gaseous products. Now question is may come that which particular these things gaseous products are available due to this carbohydrate digestion in ruminant species. It is the methane and carbon dioxides. So this methane production, this is an inevitable process. That means you cannot completely stop that methane production. So whenever there will be reduction of carbon dioxide by hydrogen, there will be methane productions. There are several microorganisms or archaea which is responsible for methane productions. They are present in the rumen, so they are basically responsible for methane productions. Okay. So along with that volatile, so what are the three major volatile fatty acids present or product produced due to carbohydrate digestion? You have to remember these things: acetic acid, propionic acid, and butyric acids. Other than that, definitely there are some other short chain fatty acids are also produced, like that of valeric acid, isovaleric acid, 2-methylbutyrate, that is there, but major volatile fatty acids are acetic acid, propionic acid, and butyric acid. So from here you can easily understand the difference between the ruminant and non-ruminant regarding the carbohydrate digestion. In case of non-ruminant, it's the glucose, fructose, galactose, that is the monosaccharide units, ultimate end product. But in case of ruminant species, it is the volatile fatty acids plus some short chain fatty acids plus gaseous products like that, methane and carbon dioxide. This bacteria, protozoa, fungi, they are the normal inhabitant in rumen. You have to remember, sometimes you may get questions also, what is the number of bacteria present in rumen content. So it is 10 to the power 9 to 10 to the power 11. There are range. Bacteria content in the rumen liquor is that 10 to the power 9 to 10 to the power 11 per ml. 1 ml of rumen liquor contain 10 to the power 9 to 10 to the power 11 numbers of rumen bacteria. And protozoa, it is 10 to the power 5 to 10 to the power 6. Even fungi also, if you can remember, 10 to the power 5 to 10 to the power 6 per ml, it is also okay. Other than that, there are some archaea, that is the methanogenic bacteria, and some bacteriophages are also there. Simply here you just remember bacteria, protozoa, and fungi, they are the major inhabitant in the rumen, and they are completely anaerobic in nature, and the number of bacteria is 10 to the power 9 to 10 to the power 11 per ml of rumen liquor. Protozoa 10 to the power 5 to 10 to the power 6, and fungi also you can remember 10 to the power 5 to 10 to the power 6. So this much you have to remember. Then coming to the carbohydrate digestion in the ruminants, what I was just talking, that how it is actually the these volatile fatty acids are produced in the rumen. So this is the schematic diagram. Sometimes you may get the questions, write down the digestion of carbohydrate in ruminants. Simply you cannot say these things that all the carbohydrates, whether it is the monosaccharides, disaccharides, polysaccharides, all gets converted to volatile fatty acid. But how it happens? This is the schematic diagram, that how the cellulose, starch, or any other structural polysaccharides like that hemicellulose, pentosanes, pectins get converted to pyruvate first. So cellulose, so definitely it's a structural polysaccharides. It is a homopolysaccharide. Cellulose will be converted to cellulose. Then this is the different pathway through which it will be converted to pyruvic acid first. Once the pyruvic acid is produced, then this pyruvic acid again get converted to different volatile fatty acids through different metabolic pathway. So here here first is the methane, that is the gaseous products, because as I have told, their gaseous products are carbon dioxide and methane. Sometimes some quantity of carbon dioxide get reduced by hydrogen to produce the methane. Other than that, there are acetate, butyrate, and propionate. So these are the three different pathways by which the three different volatile fatty acids are produced. Here, important thing which are very frequently asked question is that acetic acid, if it is oxidized, in that case, how many moles of ATP will be produced? You have to remember, there are two types of questions. One question is that what is the total quantity of ATP produced? It is 12 ATP from 1 mole of acetic acid. But if you are asked that net ATP productions, it will be 12 minus 2, because 2 is already going to be used to produce the ATP. So you have to remember net ATP production from 1 molecule of acetic acid is 10 ATP. From 1 molecule of propionic acids, you may get 17 or 18. From butyrate, from 1 molecule of butyric acid, you will be getting 25 moles. That means butyric acid, 1 molecule of butyric acid is going to produce maximum number of ATP. This is very frequently questions. You may get questions in your theory papers also. Definitely, this type of questions, whosoever, definitely this question paper which is going to be set by the external examiner from other university other than the CAU. So they used to ask this type of questions, that from 1 molecule of acetic acid, how many moles of ATPs are produced? So you have to write 10. So if you are asked for the butyric acid, it will be 25 moles. If it is propionic acid, it is generally 17 to 18. Then another important thing is that out of the 3 volatile fatty acid, which one is considered as glucosinic volatile fatty acid? From the name itself, you can understand glucogenic means it it can synthesize glucose. Yes, it is the propionic acid. So out of these three volatile fatty acids, only the propionic acid is glucogenic in nature. That means it can synthesize glucose. Is it clear? Are you getting me?
Yes, sir.
Yes, sir.
So here, this ruminant species, do you know what is the glucose level in case of ruminant species? It is hardly 40 to 60 milligram per 100 ml. That means the quantity is very less glucose level in the ruminant species, but most of the glucose which are there in the blood circulation, the ruminant species for cattle and buffalo, it is from the propionic acid only, maximum. But definitely some glucose are also produced, some ketone bodies are also there. So because in the central nervous system, they need the fuel from glucose, even sometimes from the ketone bodies. So definitely some glucose is required to be produced, and that particular role is performed by the propionic acid, because from the propionic acid glucose can be synthesized. Then another important questions which are generally asked: when these volatile fatty acids are produced in the rumen, what will be the fate of that volatile fatty acid? Definitely through the ruminal wall, that particular three different volatile fatty acid will cross the ruminal wall, and that will be there in the blood circulations. Now question is there whether they are crossing the ruminal wall as such, or there are some changes in the ruminal wall itself. Yes, there are some changes in the ruminal wall. What are the changes? Acetic acid is not going to be converted to any products or any substances, but the propionic acid, it is going to be converted to pyruvic acid and lactic acid. 50% of the propionic acids are going to be converted to propionic acid and lactic acid. 80% of the butyric acid is going to be converted to ketone bodies. You may be asked, what are the different ketone bodies? Acetic acid, beta hydroxybutyric acid, and acetoacetic acid. So beta hydroxybutyric acid, and butyric acid, beta hydroxybutyric acid. And acetate is not going to be metabolized as such. It is going to be there in the blood circulation as such. So question is asked, which particular volatile fatty acid is found in appreciable quantities in the blood circulation or peripheral blood circulations in ruminant species? You have to answer it acetic acid. Why it is acetic acid? Because acetic acid does not go for any sort of metabolism in the ruminal wall. And that's why, for the milk fat synthesis, acetic acid is the major precursor, because it is found as such in the blood circulations, and the mammary gland cells can easily uptake that particular acetic acids, and it is used for the synthesis of the milk fat. So from there you can understand propionic acid 50% metabolized in the ruminal wall itself to propionic acid, this pyruvic acid and lactic acid, and this one butyric acid to ketone bodies, and acetic acid as such it cross the ruminal wall. And here these things already you have studied in details in your biochemistry class. I'm not going to tell in details. Simply you have to remember glucose, if it is oxidized in the cell by glycolysis process, what will happen? Glucose will be converted to 2 molecules of pyruvic acid. Then what will happen? Again, it will enter to the TCA cycles. Then in the TCA cycles, what will happen? That 1 molecule of pyruvic acids will be producing 15 molecules of ATP. So as you have seen in the glycolysis, 1 molecule of glucose is producing 2 molecules of pyruvic acid, and each molecule of pyruvic acid is producing again 15 molecules of ATP through TCA cycles. So you may be asked the questions that if 1 molecule of glucose is completely oxidized in the presence of oxygen, that means in aerobic conditions, how many molecules of ATP is going to be produced? Total ATP productions I'm talking. So there will be 8 molecules from glucose to pyruvic acids in that glycolysis cycles, and 15 into 2, that means 30 molecules in the TCA cycles. So it will be 30 plus 8. So ultimately what will happen? There will be production of carbon dioxide, water, and 38 molecules of ATP. So you have to remember that 1 molecule of glucose will be producing 38 molecules of ATP plus carbon dioxide and water. Other than the two important cycles which I have told, glycolysis and TCA cycle. Glycolysis occurs in cytoplasms and TCA cycles, that is the Krebs cycles, it is in mitochondria. Now, other than that, one more cycle is metabolic pathway is also involved for the metabolism of glucose, that is the HMP, hexose monophosphate pathway. You have studied in biochemistry also. Here actually basically you are not going to get any ATP, but it is also very important metabolic pathway. What for it is required? Here you need to remember all this pathway for the HMP, that is maybe required for your biochemistry, but for nutrition you have to remember HMP pathway is required to produce two important metabolites. What are they? One is ribose, another one is the NADPH. What for this ribose is required? Ribose it's a monosaccharide having 5 carbon atom units. So this ribose is required for the synthesis of DNA and RNA. So this ribose is produced over here. Another one is the NADPH, nicotinamide adenine dinucleotide phosphate hydride. This NADPH is required for the fatty acid synthesis. So basically through HMP pathway, glucose is producing ribose and NADPH, which is required for, this one is NADPH is required for the fatty acid synthesis, and ribose is required for the DNA and RNA synthesis. This much you should remember. You need to remember whole pathway. Okay. Then another important thing is that you might have heard these things, terminology, that is gluconeogenesis. Gluconeogenesis means it is the production or synthesis of glucose from non-carbohydrate sources. So that non-carbohydrate sources may be amino acids, maybe glycerol, maybe volatile fatty acids also. What is that volatile fatty acid? Just right now I told these things, that is the propionic acids. So you have to remember through gluconeogenesis process only propionic acid get converted to glucose. So, and one more important thing you have to remember, gluconeogenesis is not just a reverse pathway of glycolysis. Some of the paths are there where there are requirement of separate enzymes. I'm not going in details about this biochemistry. In that case, definitely this class will be over just to discuss this glucogenesis. Here, only thing you have to remember, propionic acid get converted to glucose, gluconeogenesis process. And these propionic acids, here actually questions are asked, when propionic acid get converted to glucose, what are the different vitamins required for that conversion of propionic acid to glucose? You have to remember two vitamins are required for conversion of propionic acid to glucose. One is biotin, another one is vitamin B12. You have to remember these things, propionic acid to glucose, because you know this is the different pathway. Propionic acid get converted to succinyl CoA. Once the succinyl CoA is formed, it will be entering into the TCA cycles. Then it will be producing via the reverse pathway glucose. So two important vitamins, one is the biotin, another one is the vitamin B12 for the conversion of propionic acid to this glucose. And some important point also, very frequently asked questions which you have to remember for your objective type of questions. One thing is that what is the total quantity of volatile fatty acid production? It is 4 kg per day. Okay, and this is the non-glucosinic ratios. I'm not going to discuss these things. Non-glucosinic ratio is acetic acid plus 2-butyrate plus valeric acid divided by propionic acid plus valeric acid. This is the non-glucosinic acids. Questions are asked, what are the different vitamins required for the methane productions? Methane productions require folic acid and vitamin B12. Here also, once I am talking about the methane productions, two important points you just note it down. The calorific value of methane is 13.34 kilocalorie per gram. Calorific value or gross energy value of methane is 13.34 kilocalorie per gram. One thing, What are the vitamins required for methane production? One is the folic acid, another one is the vitamin B12. Another important thing is that the methane, if 100 gram of digestible carbohydrate is there in the rumen, how much quantity of methane is produced? Very frequently asked question. It is 4.5 gram. 100 gram of carbohydrate, if get digested, there will be productions of 4.5 gram of methanes are produced. That is important questions. Then whether all the monosaccharides which are produced due to the digestion of carbohydrate in non-ruminants, they can be absorbed at equal rate. It is not like that. Rate of absorption of this glucose, different molecules like that. Galactose absorbed faster, followed by glucose, then fructose, then pentoses. Then efficiency of free energy capture from 1 molecule of glucose is 69%. What does it mean? That whatever energy is produced due to the oxidation of glucose, you should not think that all the energy is captured in the form of ATP. It is not like that. 31% of that energy is lost as heat. Hardly you will find that 69% energy is going to be captured in the form of ATP. Okay, and the methane production, as I have told, it's a inevitable process, but basically it is the wastage of energy. You may get the questions that how much quantity of gross energy is lost as methane. Very frequently asked questions, that is 7 to 9% of the gross energy is lost as methane. So this is in brief about the carbohydrate classifications, digestion in ruminants and non-ruminants, and their metabolism part. If you have any query about the digestion of carbohydrate, you can ask me. Otherwise, I will switch on to protein digestion. Any query? Okay, should I go for that protein digestion? Should I go for next one?
Yes, sir.
Okay.
Yes, sir.
Wait. Well, is it visible? Okay. Now, hurriedly, I'll be going through that protein digestion part. So here, what you have to remember, first important questions which are asked about the amino acid classifications. So amino acid classification, you have to remember the so first one is the monoamino monocarboxylic acid. What does it mean? There will be one COOH group and one amino group. That means it will be the neutral one. So neutral amino acids. Another one is the monoamino dicarboxylic acid. There is two COOH group, means definitely it will be acidic in nature. So it will be the acidic amino acids. If there are two amino groups, it will be considered as diamino monocarboxylic acid. So definitely it will be the basic amino acids. There are some amino acids which does contain sulfur. There are some amino acids which are aromatic in nature. Then there are some heterocyclic amino acids. There are some amino acids which are formed from the special sources. So here, hurriedly remember these things. Yeah. So these are the neutral amino acids: glycine, alanine, serine. You see, there is one carboxylic group, one amino group. Then, other than that, here the monoamino dicarboxylic. This is also threonine, valine. Here, Yes. Monoamino dicarboxylic, that means it is the acidic amino acid. You have to remember what are the acidic amino acid: aspartic acid and glutamic acid. These are the two amino acids which are considered as acidic amino acids. Otherwise, it can be termed as monoamino dicarboxylic acid. Okay. Then diamino, that means there will be two amino groups. That's why it's basic amino acids: arginine, histidine, lysine. These are the three amino acids which are basic amino acid. Very frequently asked questions: name some basic amino acids, name some aromatic amino acids, name some acidic amino acid. Simply you have to find out the correct answer from the objective type of questions like that, multiple choice, or you have to fill it fill in the blanks. So basic amino acids, that is the three amino acids: arginine, histidine, and lysine. Aspartic acid and glutamic acid, they are the acidic amino acids. Then sulfur containing amino acids, it is all known to you, that is the cysteine, cysteine, and methionine. So two molecules of cysteine that will be combined together to produce this cysteine and methionine. All these things are sulfur containing amino acid. Then aromatic amino acids: phenylalanine and tyrosine. Then another one, these amino acids, again, it can be classified based on their metabolic functions. Metabolic functions means if the amino acids are required for glucose synthesis, they will be termed as glucosinic amino acid. If it is required for the ketone bodies formation, they will be ketogenic amino acids. And some amino acids are there which are having both the properties. I mean, it can produce glucose or it can produce some ketone bodies also. They will be considered as both glucogenic and ketogenic. The question which is generally asked is that which particular amino acid is considered as true ketogenic amino acids. So true ketogenic amino acid is leucine. You should not forget these things, that leucine is the true ketogenic amino acids. Other than that, all this isoleucine, lysine, they can also produce ketogenic acid, but only leucine can produce only ketone bodies. That's why it is considered as ketogenic amino acid. Then question is asked, what is isoelectric pH? Isoelectric pH means this is the pH value for a given amino acid at which it is electrically neutral. So that is known as isoelectric pH. Then another important thing regarding the protein classifications. Every time you will find that some of the questions will be asked regarding the ketogenic, classification of protein. Simple protein, globular protein, and there are some derived protein. So simple protein, another simple protein, again globular protein. What are the different examples of globular protein? Albumin, globulin, prolamins, histones, protamine, all are the example of globular protein. Then what are the this one fibrous protein? Fibrous protein examples are collagen, elastin, and keratin. Okay. Then conjugated protein. Conjugated protein, on hydrolysis they will be producing amino acids plus one there will be non-protein fractions which is known as prosthetic group. So these things, based on the prosthetic group, there are several conjugated protein. One is the nucleoprotein conjugated to the nucleic acid. Metalloenzymes, they are having some metals as an integral component. Phosphoprotein conjugated with the phosphoric acid like that casein, phosphatin, lipoprotein with lipid. So all these are examples of conjugated protein, because along with the amino acid fractions there are some non-protein fractions which is known as prosthetic group. Then another one is the derived protein. It may be primary derived protein, it may be secondary derived protein. Okay. Then along with that protein structure, we usually discuss about the non-protein nitrogenous compound. From this non-protein nitrogenous compound also you may find one or two questions. What are the different non-protein nitrogenous compounds which are having immense animal nutrition significance, particularly for the ruminant species? It is basically the urea and biurate, but mostly it is the urea. So urea is considered as in non-protein nitrogenous compounds. Why it is important for the ruminant species? Because ruminant species have the unique capability to utilize this non-protein nitrogenous compound and get it converted to microbial protein, which is having excellent amino acid compositions. So most of the animals, if they are not getting good quality protein, they can utilize this non-protein nitrogenous compound in the form of urea, and it can be converted to microbial protein. So question is asked whether the single amino acids, let it be lysine, lysine is considered as protein or it is considered as non-protein nitrogenous compound. You know, if you can remember the definition of protein or true protein, that is the polypeptide chains where the amino acids are linked by peptide linkages. So if amino acids are linked by peptide linkages, they know which protein. You should remember it is coming under the non-protein nitrogenous compound. Non-protein, that means it is not a protein, but it does contain nitrogen as an integral component like that of protein. That's why it is termed as non-protein nitrogenous compound. Now, question is that how this urea gets converted to microbial protein. 2-3 important points you have to remember. What will be the quantity of urea to be added in the ruminant's diet for the utilization, optimum utilization? 1% of the total diameter intake. If the animal is consuming 10 kg diameter, 1% of the diameter should be through urea. You should not go beyond that one. Another thing is that if you want to mix this urea in the concentrated mixture, it should be 3% of the total concentrated mixture. Suppose if you are preparing 100 kg concentrated mixture, maximum you can add 3 kg in the concentrated mixture. And sometimes this urea is utilized for the improvement of nutritive value of poor quality rappages, like that straw. You know, straw does not contain any digestible protein, and the protein content is very less, hardly 3.4%. But if you are adding this urea, or if you're treating that poor quality rappages with this urea, the protein content or crude protein content is going to be more. So what will be the quantity? It will be almost, you can say, 8 to 9%, and the digestibility is also going to be improved also. That's why urea can be added to improve the nutritive value of poor quality rappages. Then in that case, how much urea can be added to improve the nutritive value of poor quality rappages? It is 4%. So 4% and 40 liters water is also required. So 3 things I told. 1%, it will be based on the total diameter consumption maximum. If it is mixed in the concentrated mixture, it will be 3%. If it is used for poor quality rappage treatment for improving these things, nutritive value, it will be 4%. That 3 different value also you have to keep it in mind. And one important aspect is that what is the crude protein content in urea? You know, urea content almost 46% nitrogen, 46.1 or 46.2. So 46% nitrogen means if you have to convert this nitrogen to crude protein, what we have done in the practical also, that it is to be multiplied by 6.25. So now you see urea does contain 46% nitrogen. That means 46 into 6.25, it will be coming around 282% crude protein. So 100 gram of this urea will be having 282 gram of crude protein, because the nitrogen content is very, very high. Then coming to the different enzymes responsible for digestion of protein in case of non-ruminant species. These are the enzymes that is in the stomach, other than the intestine, all these you have to remember. And sometimes you may get the questions that write down the this one, action of protein in known species. During that time, you have write all the enzymes which is responsible for digestion, starting from stomach. For carbohydrate digestion, I told you the digestion update takes place mouth, it will be up to that large intestine. But in case of protein, it starts at stomach level, then up to intestine level. There will be several enzymes which is responsible for protein digestion. I am not going in details because time is short. So another important aspect of the amino acid metabolism, there are two reactions. One is the transamination reactions, another one is the deamination reactions. Transamination reactions, where the amino group is exchanged between the amino acids and keto acids. So amino acids will be converted to keto acids and keto acids will be converted to amino acids. Transamination, that is the transfer of amino group. And here the frequently asked question is that what are the two important amino acids which are not taking part in the transamination reactions? This is lysine and threonine. They do not take part in transamination reaction. You have to remember these things. And one more important question which is asked, that which particular vitamin is required for the transamination reactions? That particular vitamin is vitamin B6, that is the pyridoxines. So what else is very much required for the transamination reactions? Then deamination reactions, it is simple, the removal of the amino group. So here it will be the deamination reactions. Then ruminant protein nutrition, important aspect also I want to discuss, but it is already 10:59. I think you have the genetics breeding class also. Is it not? Hello?
Yes.
Yes, sir.
So I think teachers are waiting for your genetics breeding class. You will let me know that these are required in the next week. If it is required, you will inform me through your class representative, and you will tell me that what are the specific chapters needs to be revised. Accordingly, I will take the class for these revision classes. Is it okay? Anything else you want to ask?
Sir.
Yes.
Good morning, sir. Sir, in practicals,